see physics, organic chemistry, pharmaceutical chemistry, applied statistics, mathematics, and we have awarded more than 50 PhDs in the discipline of science, and around 100 research scholars are pursuing their research, and we are striving hard. And as our university was established in the year 2006, we will be doing our level best to be topmost next to Usmania and Kakatiya in the state of Telangana. And other than this, we have got faculty who are expertised and experienced in their own field, and they're also doing projects of DST and DBT, and they're also holding patents. And now I welcome all the research persons who are with us, who are expertised in their field, and they will be with us for 2 days and deliver talk in the field of biochemistry of proteins. And I welcome you all, sir. With these few words, I would like to conclude, and I wish to congratulate convener and also coordinator who will be with us for 2 days. And Haleem Khan sir has taken initiation in organizing this program, and I wish that the workshop will be grand success and all the participants, especially students and faculty, would be benefited. And please make a note, and if possible, if needed, you also can take few points, and you need to understand and you need to stay on the platform without missing any points. And wish you all the best, wish you all the success, and thank you. Thank you, sir. Over to you, sir.
Thank you, ma'am, for your address. Now I request convener of this lecture workshop, Muralidhar sir, to address the inaugural session.
Thank you, Dr. Haleem Khan. It's my duty to explain the context of this workshop, both for the online nature as well as the very content of this workshop. This is an educational problem. All of you are aware of this. There are about 1,000 universities of different varieties and 40,000 undergraduate colleges in our country. If you remove top 100 colleges and top 50 universities, in the rest, the teaching is not very good. Therefore, generations of students are being deprived of proper guidance because of poor level of educational training. The Indian Academy of Sciences, Bangalore, came into picture and thought that it will do something in the form of supplementary education. And they formed an education panel on that about 20 years back. This education panel decided on a couple of activities to help colleges, universities in the semi-rural sectors of India and to give what we call as supplementary education, and requested fellows, elected fellows, to help in this organization. There are 2-3 types of programs that the Academy initiates and now running very well. The other 2 academies, one in Delhi, another in Allahabad, Prayagraj, they also chipped in, and so it is now a combined program of the 3 national science academies. The first program they initiated was in the form of summer research fellowships to second-year BSc students, second and third year MBBS, BTech, first year MTech and MSc students of our country. They offer fellowships. About 1,500 fellowships are offered in the area of life sciences. We get about 15,000 applications. Carefully, these are chosen, and the program makes sure that each one of these students from semi-rural area goes and stays for 3 months, 2 to 3 months, in a very advanced laboratory, say in Delhi or Bangalore or Madras or Calcutta or some such place. We also make sure that students from one part of the country are obliged to go and stay in another part of the country, serving cultural understanding as well as integration. The second program, this program has been running for the last 20 years. It's very successful. The second program they have initiated is in the form of lecture workshops and refresher courses, different from what normally the academic staff colleges of respective colleges and universities conduct. These lecture workshops and refresher courses are initiated, convened by elected fellows. They choose a local coordinator to do the legwork. And I'm extremely happy that Dr. Haleem Khan, by sheer dynamism and his commitment and passion, has bagged few lecture workshops and refresher courses. I had the fortune to participate in all of them. The one which was convened by Dr. Manohar Achari, and recently also a refresher course where Bangalore Agriculture University professor, well-known evolutionary biologist, also convened. Now, the purpose of these lecture workshops and refresher courses is not to do the same thing that universities are accustomed to do, like having a title, recent advances in life sciences or something. There is no direction, there is no theme. This has never succeeded in the university sector. All of you know the truth. We have also conducted like that in Delhi University, in Usmania, in many other places. Therefore, these lecture workshops and refresher courses are conducted on chosen themes, and carefully selected speakers come and give almost like classroom lectures, so that this will form an additional component, if not a only component, of teaching and making sure students understand these advances. And therefore, these lectures given by distinguished speakers are not research seminars. They are classroom lectures. We even call them as foundation lectures in biology, so that they're talked at a conceptual level and the students enormously benefit. I would advise even junior faculty to attend. They will also get benefit out of this. And today's workshop is one in such series. Like this, the Academy has done a great service in supplementary educational efforts to improve the standards of our country. The NIRF rankings, as well as the global rankings, all of you are aware of. There may be pitfalls in all these rankings, but at least it will tell a simple truth that there is much to be desired in the teaching standards, research standards of majority of our universities and colleges. And therefore, I congratulate Telangana University, Dr. Haleem Khan, Professor D. Ravindra Garu, Honorable Vice Chancellor, to have accepted this and then come forward for organizing this workshop. I hope in the next 2 days, all of us will get benefit by hearing to distinguished people who have made great efforts to attend this. They're all very busy people. Professor Rita Singh is here, and in few minutes, Professor Yogendra Singh will join. Dr. Haleem Khan himself is given to give a lecture on fungal allergens. I'll also chip in with a couple of lectures. So let us hear them. And one thing that I wish to say, that the audience can ask any question they want. As we know, and we are fond of telling, there are no stupid questions, there are only stupid answers. And therefore, be courageous and ask the speakers. They will explain. They will be too happy to explain your doubts and other things. So I congratulate once again. Unfortunately, I would have loved to come face to face because I miss the faces of the audience. But then this COVID has demoralized everything. We have no choice but to go through this online mode. Once again, thank you, Vice Chancellor Saab. Thank you, Professor Aruna, Professor Vidya, especially Dr. Haleem Khan, for coming forward to organize this workshop. I fully hope that, welcome, Professor Yogendra Singh.
Thank you. I joined actually long back.
Yeah, I noticed it now. Anyway, congratulations, and let us hope we'll get a feast in the next 2 days. Thank you, Dr. Haleem Khan.
Thank you, sir. Now I request our Honorable Vice Chancellor, sir, to address the gathering. Over to Vice Chancellor, sir.
respected Muralidhar Garu and Yogendra Singh and Jalendra Wagner and Naresh Thalla and Professor Rita Singh, our esteemed colleagues Professor Aruna and Professor Vidyavati, our local coordinator Dr. Ahmed Abdul Haleem Khan. So I congratulate our Haleem Khan to organize such a event recognized by Science Academies is a virtual lecture workshop and is supported by Indian Academy of Sciences, Bangalore, Indian National Science Academy, New Delhi, the National Academy of Sciences, India, Prayagraj. So I am very happy to give inaugural address in this occasion. So I request Yogendra Singh and Muralidhar sir and Rita Singh visit our Telangana University. So we are warmly welcome, Madam and sir. And today, concerning this workshop, it gives immense pleasure to welcome the convener, Professor Muralidhar Garu, resource person, esteemed delegates, staff, scholars, and students joined the virtual lecture workshop organized by the Department of Botany, supported by Science Academies. Is especially focused on proteins. Proteins were first described by Dutch chemistry scientist Johannes Müller and named by Swedish chemist John Jacob Berlin in 1938. And the protein was recognized as a distinct class of biological molecules in the 18th century by autonomy. And actually, my research work based on nanotechnology. Particularly, we are doing several work on drug delivery application by using magnetotherapy. And a part of the work carried out on especially for spintronic and iPad, laptop memory devices, and part of work carried out in the field of chemistry also, particularly in inorganic materials. So what we'll do, basically we are focused on drug delivery means we find out the cancer patient and move the magnet and inject the magnetic nanoparticles where they can identify the cancer tumor. That is death by usually using magnetic therapy technique. So 80% are experiments are succeeded, and part of the research work published in elsewhere journals in international journals like Solid Communication is research based on the materials, and other aspect in Journal of Magnetism Magnetic Materials, and the other one is nano bio interface. Is a basically collaboration research between India and abroad, and we are published bio interfaces chemistry also there, partial work chemistry and partial work from biological sciences. So that give highly impact factor. And as an editor also very impressed, particularly with the type of research work. So I welcome all the Muralidhar sir, Rita Singh and Yogendra Singh, Jalendra Wagmar to visit our Telangana University and make interaction with our research students, and particularly our colleagues, Professor Aruna and Professor Vidyavardhini Madam, and also suggest conduct a physical conference in our university shortly with the help of Muralidhar sir and Rita Singh and Yogendra Singh also. So please come and visit, and we will discuss a particular date, and we will call some of the international scientists, and we will also support for financial assistance also. Whatever requirement generally we will provide, sir. So I wish and bless the participants of this workshop for achieving knowledge from the stalwarts of Indian Science Academy. So thank you very much, giving Haleem sir for giving this opportunity for in this inaugural address.
Thank you very much for your kind blessing, sir. Now it is a time to express vote of thanks to wind up the inaugural session. So in this early morning, we have august audience and the resource persons from different parts of our country, like capital city Delhi, we have 2 resource persons, and one is from our own Hyderabad. So I express my deep sense of gratitude towards the esteemed speakers, esteemed Honorable Vice Chancellor, Dean Faculty of Science, and esteemed our head department for joining and addressing the gathering. With these few words of thanks, I hereby declare that the inaugural session comes to an end, and we are going to begin our session 1, where we are having our esteemed speaker, Professor Yogendra Singh, sir. Sir, can you start sharing your screen?
Yeah, just a minute. So
I request Arun.
yeah, I'll do it. Thank you very much. I would like to thank Professor Ravinder for saying very nice words, and Dr. Muralidhar, who organized this program of lectures of 2 days. For I hope it will benefit the students. And thank you, Dr. Khan, for all the efforts. I remember you mailed so many times, and finally everything is there. And so my slides will be, I'm going to upload the slides. My slides will be very,
Aruna,
can you hear me?
Yes, sir. Yes, sir.
Okay. So my slides are very filled.
Yes, sir. I'm ready, sir.
But I'm sure they will be useful for many of the slides are self-explanatory. So I will also mail these slides. And again, thank you everybody for organizing this. And I hope this will be very useful for the students. So I'm going to upload the slides.
Sir, just a minute, sir. Let me take this chance to introduce you, sir.
Madam, once again. Session will be chaired by Professor M. Aruna, Dean Faculty of Science. Now I request Professor Aruna ma'am to introduce the speaker for the audience. Or to Aruna ma'am.
Yes. I deem it as a privilege to introduce Professor Yogendra Singh, who is Professor of Department of Zoology from University of Delhi and Dean Research at University of Delhi. And his areas of specialization are bacterial pathogenesis, bacterial protein toxins, signal transduction in bacteria, and specific area of research interest in anthrax and tuberculosis. And he has done a lot of work in these areas of anthrax research and tuberculosis. And he's also recipient of honors and awards, few of which are, he has received the award of President from Association of Microbiologists of India during 2020 to 2021, and Professor Appaji Rao Best Mentor Award, and J.C. Bose National Fellow Award, and Mosilio Chantier Distinguished Service Award of American Society for Microbiology, and Fellow of Indian National Science Academy, and he's also Fellow of Association of Microbiologists of India, and he's also Fellow of Indian Academy of Sciences, and he's Fellow of National Academy of Sciences, and he has received Goyal Prize in Life Sciences and All India Biotic Association Award, and also Gold Medal in BSc. He's recipient of Gold Medal and Rockefeller Career Development Award. So these are a few of the awards which has been received. And coming to his professional activities, he's editorial board member of Journal of Biological Chemistry. He's editor in M-System ASM Journal, and is reviewer of molecular microbiology. And he has got collaboration with many bodies, like collaboration with Professor Anil Tyagi, and collaboration with Dr. Stephen from USA. And also he's a visiting scientist of Max Planck Institute, Germany. And he's a CSR DAD Fellow at University of Germany. And he's a visiting special volunteer USA. Visited as collaborating scientist under CEFI PRA program in University of France, and visited as scientist in Konkuk University, South Korea. And coming to his activities in the society, he's a member of Society of Biological Chemists, life member, and Association of Microbiologists, life member, and Indian Science Congress Association, life member, and life member of Association of Clinical Biochemists, and also of American Society of Microbiology. And he's a member of committees like DBT member, and member in Special Committee of Center for Molecular Medicine, Research Council member, CCMB, and member of committees for CSIR celebrations for the Foundation Day, and also member of Selection, Assessment and Promotion Committee. And he's also advisor for Star College project of University of Delhi. And he's also member of Advisory and Monitoring Committee. And he has served as member and chairperson of Purchase Committee. And he's also coordinator for CAG audit. And out of these, coming to his activities for the department, he has got patents granted from anthrax toxin fusion proteins, nucleic acid encoding same. And this was awarded in the year 1997. A novel protein molecule useful for inhibition of anthrax toxin, and diagnostic assays for the detection and identification of aspergilli field filed on August and was in the year 2010. And out of these, he has also awarded PhDs, and count of his PhDs, you just see, imagine, number is around 53 who got awarded. 53 PhDs got awarded under his guidance. So we are very grateful to you, sir. And currently, few students are working for PhD degree, and most of his papers got selected to be at international reputed journals. And with these few words, I welcome you, sir. And sir will be delivering a lecture on the topic. Yes, topic is biomolecules and macromolecules, amino acids, peptides, and proteins, primary structure of proteins, and separation techniques and classification. So all this is dealt by Professor Yogendra Singh. And on behalf of the department, I welcome you, sir. And thank you, sir. Haleem Khan, sir, over to you.
Thank you, Professor Madla, for a very elaborate introduction.
Thank you, sir.
I'm a very common person like all of you. And maybe I will again like to thank Professor Muralidhar and Dr. Haleem Khan and Vice Chancellor sir for allowing and organizing all this program. So maybe I'll now upload my slides without wasting any time, and then I'll start the. And you have already declared the title: biomolecules, macromolecules, amino acid, peptide, proteins. So in addition to primary structure, I'm going to also talk about secondary and tertiary and quaternary structure also in this session only. Afternoon session, I will talk about modifications, post-translational modifications. Also, I'd like to tell you that today we are having some problem in electricity and sometimes so please bear with me if in case there is any problem. I will try to as soon as it comes back. But so far it is good. I hope it runs like this. So I'm going to upload my slides now. Thank you. Zoom. उसको बुला लें ये डेस्कटॉप पे था पासवर्ड क्या डालना है खुला क्या है
नहीं वो करना होगा सर आपको ना अलाउ करना पड़ेगा इसमें
क्या अलाउ करना पड़ेगा
ये जो है ना जूम के लिए स्क्रीन शेयर करने
उन्हें फोन नहीं करो मशीन वाला ने बल्कि वो तमिल लोग नहीं रहो क्यों सान
कंप्यूटर
Hello, Dr. Muralidhar. Can you hear me?
1 second. Yeah, I can hear.
Yeah, there is some problem in uploading. I'm trying. Maybe I can mail to Dr. Khan.
Yeah. You can immediately mail it. He will project from the center, his department.
Yeah.
Then you can keep talking. No problem. You have his email address?
Yeah.
Yeah,
The
please
one
send.
he mailed yesterday.
Okay.
I don't know why it is not getting.
No, it's always happen. Don't worry. I have already sent all my slides. Because any hitch comes, they will immediately start projecting.
So I have already mailed the
wonderful.
this class and he can upload if possible.
Dr. Haleem Khan?
Can he check his mail?
Yeah.
Sir, I'm doing that.
Yeah, thank you.
फॉरवर्ड कीजिए
करो जरा
सर इसमें आप लॉगिन कीजिए एक बार
किसको करना है
हलीम ये है ना ऊपर जो जिससे आया ये मेल
अपने आप से किया हो
ये आपने अपने आप को
अरे वो हटा दो उसको
मुरली क्या नाम है
अरे
नहीं हलीम
यहाँ से आया था ना तो हाँ इनको किसी को तो रिप्लाई किया था
उसमें ऊपर मेल में लिखा हुआ है। हाँ उसमें देखो जा करके,
ये
ऊपर सबसे जो है।
हाँ, इनको करो सर। आप एक बार इसमें लॉगिन करके देखिए।
अरे छोड़ यार, रहने दो।
वहाँ पे प्रेजेंटेशन
ऊपर होगा
ये वन हाँ वन तो हमेशा होता था, पता नहीं आज क्या हो रहा है। Yeah, I have mailed it to Dr. Haleem Khan. I hope he gets it soon. चला गया है।
सेंड
चला गया होगा।
सर आ गया, मैं अभी शेयर कर रहा हूँ।
Okay, all right, thank you. Probably they have to make co-sharing with us. I have to be added probably there, and then only I can do something. But anyway, now I have mailed the. Sometimes these problems. So got it, I hope.
क्योंकि अगर आपको वो कंट्रोल करने के लिए आपको यहाँ से
नहीं वो करेंगे ना, I will say कि change the. Is it okay, Dr. Muralidhar?
एक मिनट अभी शेयर कर रहा हूँ मैं।
Yeah, okay.
Dr. Khan is on the job.
Okay. Yeah. Sorry for this delay. So can everybody see these slides?
हाँ सर आ गया है, सभी को आ गया है शेयरिंग।
All right.
Make it full screen.
Make it, please make it full screen, yeah.
फुल स्क्रीन है सर, आप चालू कीजिए।
Okay. So again, I'm very sorry for all this technical glitch, which some problem. Can everybody see this slide? I can see only half of it. There is still some. Can everybody see this slide?
Yeah. You go ahead.
Okay, all right. So good morning, everybody, and welcome to this class. I'm going to talk about biomolecules and macromolecules and all these things. Can you change this slide now? Can you come to the second slide?
Sir, second slide has come.
Oh, it is not on my screen anyway. There is some issue, but anyway. So I will use my other computer. I hope I don't have problem of electricity in between. So all living molecules actually are made up of non-living things. Biomolecules actually are those molecules which are associated with the living molecules, as I have written in this slide. And these molecules, which are non-living, when they crosstalk with each other and function together, they can make a living cell.
अपने स्टूडेंट्स को डाले दीपक सर।
there is some voice. Can you hear me? Or
I request everybody to mute their microphones except the speaker.
so I thought there is somebody saying something. So maybe again I'll start. I was saying that all living cells are made up of non-living things. Those non-living things, when organized in a certain fashion, when they crosstalk, then the cells become live. Now, as the name says, biomolecules, any molecule which is associated with the living system is actually biomolecule. Those molecules actually are responsible for the structure. So these molecules make this structure, and also they are responsible for all the function which is performed by a cell. Now, these molecules can be small and large. I'm sure you all know many small molecules, which includes many vitamins, hormones, steroid hormones, protein hormones, metabolites, and there are many monomers. And these monomers, when joined together, they make large biomolecules. And these large biomolecules are carbohydrate, lipids, proteins, and nucleic acid. There exists very large variation in their size and their structure and even their function also. But these molecules have got some major job, and together they perform different jobs. So I will say that these molecules, which are carbohydrate, lipids, protein, nucleic acid, they do one major things, but combined together, they can perform many other control many biological processes. For example, carbohydrate and can combine with a protein or lipid to make glycoprotein or glycolipid. The lipid can combine with the protein to make lipoprotein. Now, if I talk about carbohydrate, the major job is to give energy. And I'll talk about why carbohydrates are selected for major source of giving energy. Lipids, as we know that lipids can store a large amount of energy. Proteins regulate metabolic pathway. Nucleic acid have got a storage of information. In recent time, it has been seen, although proteins are the major molecules, macromolecules, which actually regulate the function, regulate many metabolic pathway, nucleic acid also has the ability to regulate. There are many non-coding RNA, small RNA, which can regulate. This is a very new area of the research.
Dr. Khan, sorry for interruption. We can't see the slides.
No. Can you go to the next slide? So talking about carbohydrate, we also call it sugar, saccharide, glucoside, and they contain carbon and hydrogen and oxygen. If you go to the literature and read the books, internet, you will say it is a hydrate of carbon, polyhydroxyaldehyde or ketone. Now, these monosaccharide is the monomer of the sugar, which is the simplest form. And when these monomers join, it makes the polymer. And I'm sure you know that glucose is the molecule, is the carbohydrate, which is going to enter into the pathways to provide you energy. Especially it enters into the glycolysis and then goes to the, many things go to the TCA cycle. And these pathways actually, they cross-talk together. So I will say that carbohydrate has a main job of providing energy, and glucose is the molecule which actually enters, which is giving the ATP molecules or energy. Glucose is also used for storage, and these storage are, if you talk about plant, it will be starch, and among us, among human beings, it is as a glycogen. Now, while glucose is used for energy and even energy storage, this energy storage is only for a very short period, maybe a couple of days maximum, you can store energy as a glucose. But This can also be used. Carbohydrates are also used for structural purpose and for transport purpose. Many enzymes. So when the protein is synthesized, they are packaged and they are delivered. Many times these carbohydrates help in the transport also, transport of the vesicle to take the protein to their destination. For example, lysosomal enzymes, which are of course have proteolytic activity, lipid degradation activity. So all those enzymes are transported through the vesicle, and there the signal comes from the carbohydrate. So in addition to giving energy immediately, the carbohydrate is used for storage of energy, structural, as well as many other functions such as transport. Now, why nature has selected carbohydrate? The nature has selected carbohydrate because it is in abundance. It is very soluble. You can see how soluble glucose is. Therefore it can travel, it can distribute from storage. It can go from through the blood to other tissues. It can enter into energy producing pathways. And as you know, glycolysis and TCA are the major pathways where you can get energy. There are many other sugars in addition to glucose. There are many sugars which make, for example, plant will have a different kind of polysaccharide. So there are different sugars also in addition to glucose, and they make different polysaccharides. They can make, it can be converted into amino acid and lipid, and then they can all enter into pathways which is going to give energy, and they will make molecules for the structural components. Since energy, so to start with, I will say that energy is very, very important. First choice is always energy, and all these pathways talk to each other so that they can be diverted into other pathways also. Now coming to the digestion, how do we get these carbohydrates? Actually, when we eat food, the carbohydrate is digested in our stomach, but we cannot digest all the carbohydrate. There are many plant polysaccharides, for example, cellulose, and there are many other which cannot be digested by our enzymes because we do not produce them. They are complex carbohydrates and they are digested only by the microbes. Can you change the slide, please? Now, when I talk about lipids, the other, can you change the slide, please? The other macromolecule, you can say, is the lipid. Lipid, of course, is water insoluble. It is soluble in the non-polar solvent. I have written on this slide, there are examples which are triglyceride, phospholipid, sphingolipid, cholesterol, fatty acid. All these are water insoluble, and they are soluble in non-polar solvent. When we eat food, the larger lipids are degraded. They are resynthesized in our cells. In addition to what we can make fatty acids, but there are fatty acids which are also essential. I said that glucose is the major monomer of the carbohydrate which gives energy, but continuously energy is also given by the lipid also. Almost 30-40% energy comes from lipids in addition to the carbohydrate. So they work as an energy storage, and the reason is that lot of energy in terms of amount of energy can be stored into the smaller volume. That's why lipid has been selected to be stored, because they are not hydrated, so they will require less space for storage. Of course, they also work structural. I'm sure all of you know that lipid bilayer. So then they are associated with the signaling pathway, transport. Many vitamins and hormones are made, for example, steroid hormones. So these lipids can So basically the lipid can provide energy, they can work as energy storage, and they work as for structural purpose also, as I mentioned. Now, we do get fatty acid from our food, but they can also be synthesized from the carbohydrate. Acetyl-CoA, I'm sure you remember, when the glucose is converted to the acetyl-CoA, it can enter into the TCA cycle, or it can go for synthesis of the fatty acid. Can you go to the next slide, please? So proteins are polymer of amino acid. Amino acid, as the name implies, amino group and carboxylic group, acidic group. These proteins have got many roles. Actually, they regulate all the pathway. They are associated with the structures. Many proteins make those longer proteins, structural proteins, fibrous proteins, like collagen, elastin, etc., which work as a structural proteins. In addition to regulating the pathway and structural proteins, the amino acid can also get converted into sugars. If you deaminate them, if you remove the amino group, they will get converted into the sugar, so they can provide energy. They can provide energy also. They are associated. Many of the proteins are associated with the signaling pathway, especially kinasing and adding the phosphate group, and many other ways the signaling pathway are regulated by the proteins. Hormones, they are associated with the transport also. So amino acid, as the name, as I said, amino group and carboxylic group. But in addition to this, there will be other groups which you call side chain, R group. Alpha carbon, so there will be a carbon to which atom to which this carboxylic group and the amino group will be attached, and that carbon will be known as alpha carbon. Now the remaining portion of the molecule side chain is actually called R group or side chain. Many properties of the proteins actually are contributed by this side chain. For example, hydrophobicity, acidic nature or basic nature. So these side chains provide many properties to the protein. Now, so they are basically all they are long chain of amino acid, but those arrangement of the amino acid can make them structural protein, functional protein. You can divide in many ways. You can classify the proteins. For example, I have written here structural proteins, and I have written the names here because their structure is something like that which will not dissolve, which is not soluble. The folding of the protein is in such a way that it is going to be behaving like a fibrous structure, and that example comes from the collagen, elastin, keratin, etc. Functional proteins are those proteins, of course, which are soluble. Therefore, they are going to regulate the pathways. They include enzymes, there will be channels and transport, and the signaling enzymes, signaling proteins. You can also classify the protein based on their acidity or basicity. So acidic amino acid, basic amino acid, and acidic amino acid includes glutamic acid, aspartic acid, which has got additional carboxyl group, and basic amino acid, lysine and arginine, which has got additional amino group. You can also classify them, which can be converted into the glucose. So as I said that amino acid can also give energy because they can get converted into the glucose or those intermediates which are present into the TCA pathway. That's how they can provide ATP. So glucogenic amino acid, ketogenic amino acid, because they can make these amino acid can enter into the pathway to provide energy. Ketogenic will not form glucose, but they can get converted into acetyl-CoA, which of course can again provide the energy. You can also classify proteins into essential amino acid and non-essential amino acid. Essential amino acids are one which cannot be synthesized by and there are about 10 amino acids which cannot be synthesized by the human, but non-essential amino acid which you can make it from the when the TCA cycle is running, you can many molecules of the TCA pathway can get converted into non-essential amino acids. So you can make the amino acid from the sugar also. Can you go to the next slide, please? So let's talk about amino acid more and what is peptide and proteins. Normally, if you read the books, you will find that there are mostly 20 amino acids which are used in proteins. But in addition to 20 amino acids, there are many other amino acids which are present in the protein. For example, there is a amino acid called selenocysteine. This selenocysteine, I think there are about 25 proteins, and they are very, very important proteins. While the protein is getting synthesized, the selenocysteine, where selenium group is added into the amino acid. So it is like cysteine, but instead of sulfur, the selenium group is present. And this addition of selenium makes the protein very important for certain function. And it is added during the protein synthesis. So then many amino acids are modified. For example, I've given here example of a diphthomide. This is a modified histidine. There are hydroxyprolines which are hydroxylated. OH group is added to make hydroxyproline. So two amino acid will come together, and a peptide ball will be formed between the amino group and the carboxyl group, which will be known as peptide bond. And so the amino acid will keep joining with the help of carboxyl group and amino group to make a chain-like structure, which will be a peptide, depending on the number of the amino acid, or can become larger, which then it will be called protein. So normally we know that proteins are synthesized on the ribosome. They go into the endoplasmic reticulum, many of them. They get modified there. A rough endoplasmic reticulum, I'm sure all of you know. And many of them are modified, then they are packaged. They are transported to other organelles also. Many proteins are synthesized for the cytosol, which are soluble.
Sir,
I think Dr. Yogendra Singh's microphone is muted.
no, sir, he's unmute only.
Dr. Halim Khan,
Sir,
what is the problem?
sir, he lost a connection, I think.
Oh, okay.
Sir, I spoke to sir. There is some internet problem with him. He's trying to join again.
Okay, what do you suggest we do? Yeah. It has come back.
ఇంక నడుస్తుంది క్లాస్ అయిపోలేదు
Hello, can you hear me?
Yeah,
Hello.
I can hear.
Yes, I'm sorry, all the problem has to happen.
Don't bother, don't worry. You
Only
just
today.
remain cool, no problem.
Okay, so if you see my slide, I have made very.
I can see the slide, but whether others are able to see, I don't know.
Can everybody see this slide? Please, somebody give response, students.
Dr. Khan?
Sir, it is visible, sir, because I am also seeing live YouTube.
Okay.
Okay.
So,
Yeah.
all right.
Continue.
Yeah, okay. So proteins, of course, as I said, these are long molecule of many peptides. That's why when you take a protein, you can call polypeptide. And I have made a figure here. You can see the amino acid in colored round structures. And when they join, they become peptide. Up to 50 number of amino acid, you can call them peptide. And the longer ones, many peptide join, then you can call them proteins. Now, on your left side, if you see the structure of the amino acid, you will see that they vary in their structure. That is why when they join together in a with each other and make long chain, then they can give different structure because of their different side chains. If you remember, I said that amino group and carboxyl group attached to the alpha carbon, and the remaining is the R group or side chain, and all the properties are provided by the now coming to the protein structure, it's very easy to understand that if you consider all the sequence of the amino acid of the protein, it is called primary structure of the protein. This is nothing but just the sequence of the amino acid will be the primary structure. Now, as you can see in the picture here, can you change this slide, Dr. Khan? I hope you have the right slide. Yeah. So you can see that there are two ends, amino terminal end and carboxy terminal end, the COH. So it will start the synthesis to start from the amino and then the carboxylic group of this amino acid is joined with the amino group of the next amino acid, and this continuously goes on to make a protein during the protein synthesis. So that is why it will have two ends, one the amino terminal end and the carboxy terminal end. Now, secondary structure is nothing, but it is just the local structure which are formed by the hydrogen bonding between the atoms of the polypeptide backbone. That backbone where the NHCO is present, which has made the peptide bond, but not the side chain. So in the secondary structure, the side chain is not going to involve, only the functional group, amino group, and the carboxylic group are going to make the hydrogen bonding. Now, when they make the hydrogen bonding, they can come together in two ways. So because of their different structure, they can make two type of structure. One is alpha helix and one is beta sheet. So secondary structure consists of alpha helix and beta sheet. In alpha helix, the hydrogen bond is formed between this carboxyl group and the amino group, CO of the first amino acid with amino group of the fifth amino acid. So therefore, each helix will have certain number of amino acid, which I have mentioned here. In beta sheet, the different amino acid will join to the chain. As a result, they cannot make the helix, but rather two segments will come together. I have shown in the slide on the right side, bottom side. They will come on each other, and that can happen in between the two different polypeptide, or the same polypeptide can fold, and then the hydrogen bonding will happen. So it will make a beta sheet. Can you change the slide, please? So secondary structure is made, then the side chains are left, and side chains are of very different nature. There can be hydrophobic group, there can be charged group, they're negatively charged, positively charged, OH group. So that is why the tertiary structure is going to be to make the protein folded in a functional way, and that will be because of interaction of those groups. So you can say that three-dimensional structure or tertiary structure of polypeptide is actually the tertiary structure, which is formed because of the interaction between the side chain of the amino acid. Now, if you talk about how do they make this structure, many proteins can fold by themselves, but our cells have got many such proteins which are known as chaperone, and those proteins actually help in folding of the protein. Sometimes protein does not get folded, then they are sent for degradation. I'm sure that protein quality of the cell is always maintained so that the unfolded proteins are degraded by the proteases. So tertiary structure is made by the interaction between the different groups, hydrogen bonding, ionic bonding, di-dipole interaction, all these interactions, including disulfide bond, and all these bonds when are made, and I have showed in this slide on the right side down this red color ribbon type of structure, all these bonds actually are responsible for making the tertiary structure of the protein, which is actually the functional protein. Now, if you move to the next slide, the quaternary structure of the protein. So many proteins will be folded and they will be functional, but some proteins, some enzymes are very called complex enzyme, where many polypeptide are going to join, many folded polypeptide are going to join, and that is why they are called quaternary structure. So I will say that many proteins have only primary, secondary, and tertiary structure folded, and they can do the job, but some complex enzymes combine together where many folded proteins will join together, and when the same polypeptide folded join, they are called homo. It can be homodimer, homotrimer, homotetramer, but when those folded polypeptides are different, then they will be heterodimer, heterotrimer, heterotetramer, etc. So quaternary structures are also present into the cells. Many enzymes have got multiple polypeptides to do the job. Can you have the next slide, please? So basically, you can say that there are 4 levels of protein structure: the amino acid residues, the alpha helical structure, and then they are folded into by the side chain into the tertiary structure, and then the tertiary structures or folded protein can come together to make the quaternary structure. Can you go to the next slide, please? So I think we have to talk about the separation methods, and we do use separation method very often in our research. I have been working on the protein for a very, very long time. We clone genes and express the protein, purify the protein, and you will see that what kind of method we use, what are the very common methods. So if you talk about purification, proteins are required for multiple uses, therapeutic uses, for diagnostic purposes, to determine the structure, for determining the toxicity, if it is going to be used in a clinical trial or as a therapeutic purposes, to characterize the protein. So we need purified protein for many, many purposes, and there are many ways. The general principle, if you see affinity of the protein to something, based on size of the protein and based on charge of the protein. Can you go to the next slide, please? So if you talk about affinity of the protein, if you make an antibody column, for example, if you take antibody and if you link the antibody to a solid support, since antibody can bind only defined sequences, then that resin which has antibody will going to catch only the proteins having those residues. So you can make, this is called immunoaffinity. You can make a column of immunoaffinity where antibody will be attached to a resin, and this antibody will recognize only those proteins which will have those sequences. Okay, can you go to the antibody? There are many methods to link the antibody. Then very, very common method I am going to talk about, which is called metal affinity chromatography. Can you go to the next slide, please? Metal affinity chromatography is very, very common. And in this metal affinity chromatography, coordination is bond is formed between the metal ion, and we use this very commonly. Actually, my lab expresses the protein almost every other week, and they purify these proteins using nickel NTA column. So I have showed the structure of what is NTA. NTA is nothing but it's called nitrilo triacetic acid, where nitrogen is linked to 3-acetic acid. Now this nitrilo triacetic acid can be linked to a resin, and then it can make bond with this can make bond with the nickel, and this nickel can make coordination bond with the protein, with the histidine especially. So this resin can be used to purify the protein, which can have histidine residues. Now you can introduce the histidine residues using, when you are cloning a gene, the interest of a protein, then you can add the histidine residue in the protein at any end, either N-terminal end of the protein or the C-terminal of the protein, which I mentioned earlier, that proteins have got 2 ends, N and C, amino terminal and carboxy terminal. So if the protein has got those histidine, and you can hear me, isn't it? Hello?
Yes, sir, you're audible.
Okay, so this resin is commercially available. You can pack the column with this nickel NTA resin, and if you pass a protein which has got at least 6 histidine, then this nickel is going to make the bond with the histidine, and that protein will be caught and will stay in the column. The rest of the proteins you can wash away, and eventually you can elute the protein and use it the way you want. So you can actually make mutations in the gene and purify the protein. In a week time, you can make mutations to many sites, and you can purify many proteins, because these are very efficient, and most labs these days use metal affinity chromatography. Now, if you go to the next slide, please. In addition to metal affinity chromatography, since I said affinity chromatography, there are many proteins which can bind to small molecules, and those proteins, if you add to your protein, because it has affinity to the small molecules, for example, this maltose binding protein, it is a E. coli protein, but it can bind to a maltose, which is a dimer. So you can add the maltose binding protein to your protein genetically. You can take the gene of the maltose binding protein, you can take the gene of your interest of your protein, you can join them and then express the protein and pass it on the column. So this column will bind to the maltose binding protein, and your protein can get purified. And there are many ways where you can remove the maltose binding protein from your protein, so you can eventually purify your protein. Glutathione transferase is another protein which binds glutathione. So again, this protein, and we actually do use all these chromatography, nickel NTA, maltose binding, glutathione transferase very routinely. So you can say that affinity chromatography can be used in many ways, and once the molecule of affinity is added to your protein, then it becomes very easy to purify the protein to homogeneity. You can get single band protein if you use the affinity chromatography. If we move to the size exclusion chromatography, next slide. So we know that protein are of very different size. There are smaller proteins, 60, 70, 80, 100 amino acids, and protein can be very large. Thousands of amino acids can be in one protein. So therefore, you can use the property of the size to purify protein. It is called size exclusion chromatography or gel filtration. So I have made a slide here which you can see that if there are different size of protein and you pass it through column, and the resins have got hole into the holes are present in the resin, so the proteins are going to enter. So the protein which does not enter into the resin, that hole, that will pass through the column, and so based on the size, the protein will get separated, and you can purify this also quite quickly. This is also called size exclusion chromatography. Now, these chromatographic techniques are not actually absolute. You cannot purify something and say, this is the size of the protein. And this chromatography is very important for determining if there's a complex protein. That means if there are multiple polypeptide, you cannot find out, you have to use gel filtration chromatography to identify that it is a complex protein made up of multiple polypeptide. The reason is you can know that there is a gene which makes a protein. That protein gets folded to get a job or a reaction or a pathway, but you will not know whether this protein combines with other protein to do the many jobs or regulation. So I will say that to determine the size of the those proteins which make multimers, is this chromatography is very commonly used. I have been telling that packing material have got pores, and those proteins will enter into the pores which are smaller than the pore size. So therefore they will be get eluted very late, but the one which does not enter will get eluted very quickly. So this separation is based on size of the protein, but you always need the standards to determine the size of the protein. That's why you get colored standards, and when they get eluted, you can make a graph, and when your protein comes out, you can identify that this makes a dimer, trimer, tetramer, or whatever size of the protein is. Can you go to the next slide? The next slide is actually I've described that what is the stationary phase in this and what is the mobile phase. Can you go to the next slide? The third type of chromatography is actually ion exchange chromatography. Now, I mentioned that a protein, the side chains have got amino acid which can be negatively charged or positively charged. And based on the charge, you can always employ such resins which can bind the negatively charged protein or positively charged protein. And then you can use different kind of solvent which may have salt, or you can change the pH, and based on, you can change the charge and you can elute the protein. So based on the charge of the protein, you can purify the proteins, and this is called ion exchange chromatography. I think I'll stop here because I think my time also is getting over. So I will end this class, session, and we'll take the question. Or I'm very sorry for the way this
End the class.
but anyway, there are things which are not under my control. Yeah, so thank you very much. Can you go to the last slide now? I think I'm done.
Sir, one question is,
if you read I will suggest that if the student go through this slide, I have made in such a way that you will understand very easily. So these slides have been made with the help of many books and internet. So you can, if you read, this is a combined picture of what I just mentioned. Yes, that's all. Sorry about that, the way it went on. Dr. Mullin, you were saying something?
sir, what would be the cost of ion exchange chromatography setup at present?
So some resins are very, very costly, but these can be reused. But I can tell you that these days, very few people use this chromatography. Only maybe in industry they use it, but in the labs, normally we use affinity chromatography because they are very quick. Again, there also you can use the same resin. So the cost of the ion exchange chromatography, the material is costly, but you can use it many times. But more convenient is affinity chromatography, especially nickel NTA chromatography. So most labs, especially where you want to understand the mechanism and want a small quantity of protein, there the affinity chromatography is used.
Sir, the mixture, what you are going to isolate or separate, usually should be in a liquid state, isn't it?
Yes, if you are going in affinity, every place it has to be in the liquid state because it has to the resin, in affinity, the affinity material will be picked by the small molecules, as I said, nickel and T and things like that. If you are separating based on size, so it has to pass through the column. So every time you have to pass the protein in a liquid manner, in a liquid phase.
So presently chromatographies are not very much used in proteomics and all like that. Whatever facility centers are there,
Okay. When you say proteomics, it becomes very different. Okay. So normally, many of us study single protein. We mutate many amino residues in the protein, and we want to understand which residue is doing what, why it is important, what the mechanism this protein has. For example, kinases, how do they interact with their partners? So if you are purifying single protein, these methods which I mentioned are good. Now, if you want to do proteomics, that means you want to study all the proteins of a cell. In that case, you have to pass through the column where protein can get separated also, and then they will go to a machine where based on their peptide mass, they will be identified. So it is getting separated also, at the same time it is getting identified also. So LCMS you can call it. It is a liquid chromatography attached a mass machine where protein can get separated also and get identified when you are doing the proteomics.
Thank you, sir. Thank you so much. Muralidhar sir, can you start sharing the screen?
Yeah, just 1 minute. Dr. Singh,
yeah,
extremely lucid lecture. Your slides are extremely good, and I'm sure the students, after going home, please read it again. When your mind is fresh, you will understand this field very well. It's an extremely good, beautiful lecture I have heard. Thank you, sir.
Sorry the way it went, many disruption. Actually, that also.
No, that's okay. No problem. Don't worry.
So I was thinking, I have a lecture at 2 o'clock.
Yeah.
Will it be okay if I send a link? This Google Meet is very good. Actually, it gives no problem mostly.
Yeah, but these people have purchased Zoom.
Oh, okay. All right, we'll try to upload. I hope I will mail the slides again. And,
yeah. Each one of us can give our slides to the organizer.
Yeah.
So leisurely he can focus it, and the students can learn very well.
Yeah. Okay, I will do that. Yeah. So maybe I'll join again at 2 o'clock.
Yeah, 2 o'clock.
I always say very sorry again.
No. Yeah, don't be apologetic. This is
Actually,
India.
I could not tell the way I wanted to because it got so much disruption.
No. This is India. Everything is power dependent. Yeah.
So I will join at 2 o'clock again. I'll mail the slides.
Sure. Thank you so much, Dr. Sir.
Thank you very much.
Dr. Khan?
Sir.
Do you want to give a 5-minute break, or you want to start the next one?
Yeah, start, sir, now.
Okay, let me start sharing the slides.
Yeah.
Let's see what problems I have. Can you see the slides?
Yes, it is fantastic. It has correctly come.
Don't say fantastic. Let me speak.
It is like yesterday today we have tried.
Yeah.
So the session will be chaired by Professor B. Vidyavardhini Madam, Head Department of Botany. So over to Madam. Madam, please introduce the speaker.
Yeah, good morning, everyone, once again. And thank you, Halim Khan. It gives me immense pleasure to introduce the speaker as well as the convener of today's today's session as well as the whole program. It is Professor K. Muralidhar sir, who's from School of Life Sciences, University of Hyderabad. And he's speaking on the topic, evolution of functional protein, conceptual treatment, phylogenetic analysis. Muralidhar sir is a distinguished biologist of our country and was born at Coimbatore, Tamil Nadu. He obtained his BSc and MSc from Usmania University, Hyderabad. He started his research work under K.L. Ramchandran at Usmania University in the year 1969. He obtained his PhD from IISc Bangalore in 1976, working under N.R. Maugdal. His PhD work demonstrating the beta subunit of luteinizing hormone can bind ovarian receptors fetched him the Professor K.V. Giri Memorial Gold Medal for the best PhD thesis of the year. He was selected for the first lectureship in the School of Life Sciences at the newly founded Central University of Hyderabad in August 1976. During 1979 to 81, he was a research associate in the Department of Cell and Molecular Biology at SUNY Buffalo, New York, USA, and O.P. Bahal. His work led to the development of the most sensitive RIA for the HCG, a pregnancy hormone. He also demonstrated the immunocontraceptive vaccine potential of DS5-HCG beta subunit. He joined the University of Delhi in 1983 as a reader in biochemistry and became a professor in endocrinology in 1988. He was the chairman of the department during 2001 to 2004. He initiated a long range of research program on water buffalo endocrinology. His laboratory has developed procedures for indigenous production of buffalo hormones like LH, FSH, TSH, GH, and PRL, sensitive homologous immunoassays and heterologous bioassays for these hormones, besides characterizing the physiochemical and immunological features. The laboratory among the first to report the LH and PRL were sulfated, and this contributes to microheterogeneity. His laboratory later discovered the presence of tyrosine O sulfate in sheep and buffalo PRL. Using biophysical techniques, his group has studied the protein unfolding and folding of the buffalo GH. Naturally occurring size isoforms of buffalo prolactin, cathepsin derived peptides from buffalo prolactin, and a synthetic peptide based on the internal sequence of the buffalo prolactin was discovered to be powerful inhibitors of angiogenesis stimulated by VEGF or bradykinin. His group has published over 150 articles in peer-reviewed journals and books as chapters. Professor Muralidhar is an outstanding teacher and has taught courses in biochemistry, gamete biology, immunology, endocrinology, and cell biology and medical chemistry at University of Hyderabad, South Asian University, and at University of Delhi. So far, he has mentored 27 PhD and 26 MPhil research students. He has published about 150 articles in peer-reviewed international and national journals and edited numerous books. Professor Muralidhar was the chairman of the UGC Curriculum Development Committee for Zoology and brought out a benchmark report on the MSc and BSc in Zoological Sciences. For NCERT, he brought out new textbooks for higher secondary school biology in a completely new format backed by a sound philosophy. Amazon, after proper survey and referring in 2017, ranked these books at the top 5 of 750 such books in the world. He assisted University of Delhi in restructuring undergraduate science education and introduced new honors course in integrated biology, which is the first of its kind in the country. He was the convener of the NET examination and brought more balance in the examination, giving representation to all important areas of biology. He was the chairman of PSC Animal Science of DST for around 3 terms and nurtured research in the area. He has assisted all the funding agencies like CSIR, UGC, ICMR, DBT, DST, MOEF, and ICAR in various capacities. He has served as a research advisory committee of CCMB, IGIB, MRC, NBRC, ARI, and NII, besides assisting in the selection and promotion of faculty in a number of educational and research institutions in our country. He had adjudicated more than 350 PhD theses and 35 from around 35 universities and research institutions. Professor Muralidhar is an elected fellow of all the science academies of our country, that is NSAI Allahabad, INSA New Delhi, and IASC Bangalore. He has delivered the Sada Swarakshmi Endowment Lecture for SBC 1 in 1996, U.S. Srivastava Memorial Lecture for NASI in 2007. V. Gopalakrishna Rao Endowment Lecture for Osmania University in 2004. Y. Subbarao Rao Memorial Lecture for GS Indraprastha University 2007. Professor M.R.N. Prasad Memorial Institute Lecture for INSA. Hargobind Corona Memorial Lecture for GGS IP University and G.P. Sharma Memorial Lecture of Punjab University. P. Govindarajulu Endowment Lecture for Madras University. Platinum Jubilee Lecture for Indian Science Congress and many more. He served as a member of the Executive Council of NASI and INSA. He was the chief editor of the proceedings of the Indian National Science Academy for over 6 years and he had transferred the journal and brought international recognition. He was the chairman of the INSA National Institute Committee for Cooperation IUBS for a term. He was a Sir J.C. Bose National Fellow DST during 2014-16 at South Asian University, New Delhi. He was Jawaharlal Nehru Chair Professor during 2017-18 at University of Hyderabad. He was a member of SERB. He is presently on the advisory board of Inter-University Center of Teacher Education at Banaras University and a visiting professor of Annamalai, Madras and Hyderabad Universities. With his vast experience, I hope all of you have a good time and a warm welcome to you, sir. Over to you, sir.
Thank you, Professor Vidya, for a very kind introduction. Dear participants, you have just heard a beautiful lecture, a classroom lecture on proteins, basic aspects from Professor Yogendra Singh, a very distinguished scientist. I wish to today touch upon some of the controversial things and bring conceptual clarity on something that all of us are talking about, what we call as evolution. However, what I do in this lecture is to look at this evolution, one from the classical evolutionary theory of Darwin and Wallace, and then see whether this idea can be applied to nucleic acids and proteins. And what do we mean actually by this biochemical evolution? There is a lot of confusion even in textbooks. So I will try to clarify these things. So listen to me carefully. If you have any doubt, kindly ask immediately. Now to begin with, let us look at what is biology in a broad way. Biology is the science of living organisms. It is the history of life on Earth. And it is taught, of course, in different ways, in different fractions, as botany, zoology, microbiology, phylogenetic-based teaching, or functional aspects like biochemistry, biophysics, genetics, physiology, etc. So this biology, if you start from Aristotle, say 325 BC, it has grown in the last 2,100 years, let us say, in 3 phases, PHA-SES. In phase 1, it was purely descriptive. You can call it phenomenological biology. It is based on observations, either with the naked eyes or subsequently with magnifying lens and then with an optical microscope. So it was pure observation and description of whatever you see. There was no question to ask. This is called classical biology. No experiments were done till 18th century in biology. It was all observation, and it is, you can call it also natural history. This resulted in natural history catalogs and good areas like taxonomy and related aspects like ecology and evolution. Subsequently, in the 17th century, there was a French mathematician called René Descartes who advised everybody that it is not enough if you go on observing and describing. You will have to do experiments, ask questions, and try to get the answers by doing experiments. This is called experimental biology. It is also called as reductionist biology because it is reducing biology to physics and chemistry and sometimes to mathematics. Why it is called like that? There is no biological method. When you want to do experiment, you need a method. Unfortunately, till today, even Now there is no biological method. Even surgery is a physical technique. Therefore, using physical and chemical techniques, tools, biologists try to do research, experimental research in biological systems. And this is essentially in an investigative mode. And that means you start by asking a question and then get the answer by investigation. This is called reductionist biology. It is continuing. It is the most glamorous part of biology. And this is the last 350 years we are practicing. And it is all influenced by a mathematician, René Descartes. That's what is written here. This is the most dominant. You understand biochemistry, biophysics, molecular physiology, molecular biology, and all. Therefore, this is the most dominant and visible phase of biology. It does not explain, however, kindly note this sentence, emergent properties characteristic of biology. If you take, for example, neurobiology, mind is an emergent property of brain. Biochemistry or reductionist biology does not explain this. Evolution is an emergent property of underlying living processes. Biochemistry does not explain this. Therefore, reductionist biology cannot explain emergent properties. We leave it at that. Some other day we'll discuss more details. The third phase, which started about 20 years back, tired of doing experiments and trying to understand biology. The classical phase, there was no understanding. We got, of course, taxonomy, evolution, ecology, and other things, systematics. The second phase, reductionist biology, we got great progress, but understanding biology was a casualty. So tired of all this, then people wanted to know how to understand emergent properties. And so we entered third phase where people don't do experiments. They do simulations and modeling using computer techniques. So we can call this in silico biology, bioinformatics, computational biology, computational chemistry. These are all the areas under this. And it says, it believes that interactions among the components of a system leads to emergent properties of the whole organism level. This is largely practiced through simulations done with computers. So if you look at, therefore, as we said, biology is a history of life on Earth. If you look at the entire history of Earth in a very short one slide, you can make out from here that Earth is about 4.5 billion years old. 1 billion is 10 to the power of 9. Life originated about 3.5 billion years ago. This is what the scientists are telling. Of course, the religious leaders will tell something else. We will not discuss all that. The oldest fossil is about 550 million years old. Mammals, you and I belong to this. Elephant, rat, cat, tiger, all these, they originated sometime approximately 200 billion years back. The giant vegetarian mammals, 30 to 80 million years back.
Sir.
Ancestors of the great apes. Can people switch off the microphone? Thank you. Ancestors of the great apes like chimpanzee, gorilla, all this about 11 million years back. Man-like apes, Australopithecus, 4 to 2 million years back. And ape-like man, Homo erectus, and other 1.9 million years back. What we call as modern man, Homo sapiens, you can also call it as Homo sapien sapien. It does not include Homo neanderthal. And this modern man is approximately given an age of 2 lakh years, or more correctly, 50,000 years back. So you see that in such a 4.5 billion years back history, life has occupied only 3.5 billion years, and man has come only recently. But we are unique that we learn all these things through what we call learned behavior. Other organisms have instincts. I will not go into detail. We'll see that later. Again, if you look at it as a recent history, discovery of agriculture and animal husbandry approximately 10,000 years back. Therefore, man became living in social groups, and that's where intelligence developed, language developed, settled society developed, all so-called modern history developed. The Indus Valley civilization, of which you have heard of, is only 4,000 years back. The Vedic civilization is about 1200 years back, BC. Moses, the Jewish prophet, and Buddha, the man who started Buddhism, are hardly 500 BC. Alexander the Great, who came to India in 324 BC, Chandragupta Maurya, the Mauryan empire establisher, is 325 BC. Darwin wrote a book on organic evolution, 1859. We got independence in 1947, and we are now talking in 2022. In this 10,000 years, therefore, enormous progress has happened in the civilization, and we also have increased our human knowledge because we can learn much better than other organisms. So many people have established this, and the chemistry of universe seems to be leading to life. Universe seems to be in the business of creating life. This is said by Cyril Ponnamperuma. He's an exobiologist from Sri Lanka, but settled in California. He passed away long back. If you look at what Francis Crick, the person along with Watson who gave us the structure of DNA, biologists must constantly keep in mind that what they see was not designed, but rather evolved. The word design is used by religious people to say that God has designed the entire biodiversity. Crick and others and all the practicing biologists today believe there is no design, it is all evolved biodiversity. Therefore, evolution is an established fact, and the persons who initially told us about it in great detail are Charles Darwin, to whom major credit goes, but along with that, an young boy at that time, Wallace, who worked in Malaysian archipelago, he also said the same thing. I don't have time to go into detail. All this is only done for a background. So the major question that people asked biologists is, when did we start life? What is the origin of life? Is it a singular event like the origin of universe, which the physics people are telling us, the Big Bang theory and others? Chemical evolution is a term used by evolutionary biologists, but I will explain later, this is a misleading term. There are textbooks on this, but I don't think anybody understands this. I'll explain why. The cell theory of Schleiden and Schwann, which all of you have heard, is applicable to cellular forms of life only, because a major component of their statement is that a cell is a unit of life, but all cells have come from pre-existing cell. It gives a big gap as to how did the first cell come. Schleiden and Schwann has no idea about it, but this is the most popular theory which all your teachers are teaching. I'm not questioning the theory, but it gives you more ignorance than any knowledge. How, when, and where did the first cell come? Everybody is silent about it. To understand the history of life on Earth, therefore, number of people proposed a number of theories. Without going into detail, there are three types of such theories. Number one, which most of us believe, Darwin and Wallace ideas were from a single ancestor. Kindly note, single ancestor through inheritance with change and natural selection as a force, biodiversity has expanded. This is the ideas of organic evolution by natural selection. We will not go into detail. The second idea is also there. Of course, most of us don't believe it, but we'll have to give a historical fact. Idea of transformism, where separate origins of all life forms, they originated separately, but some have changed, others have not changed. So in this, there is a possibility of extinction also. There is no descent with change here. There is no branching, no adaptive significance, but only design. Then we have the third idea, which is also being circulated, that the species are always fixed. There is no evolution. There is no change. Please remember that the word evolution, the etymological meaning is change. And this third type of idea denies any change in life in the last so many years since creation. And therefore, this almost is identical to creationism of what the religions are telling. Darwin, when he said in 1859, after a 10-year gap, Darwin gave a partial answer to how did it all happen when he used the term natural selection. Others like Spencer and others added survival of the fittest. But this survival of the fittest is the most misunderstood idea. It is not what many people today talk, the sociologists and others. Here is a picture of unrelated organisms like sharks, ichthyophysis, dolphin, penguins. Penguin is a bird. Dolphin is a mammal. Shark is a fish. So you can see the body design is similar. Looking at such things, people talked about convergent evolution and other such ideas. Similarly, there are many other examples. We are not here talking about the classical evolution. And therefore, if you take this evolutionary process, it takes, as per Darwin and as per modern data, it takes millions and millions of years. I told you that 3.5 billion years back, life originated. And in this last 3.5 billion years, it has evolved into different forms of biodiversity. And therefore, the age of Earth and the age of life, they're all looked at as a big history. They're divided into epochs and eras. I will not go into detail. You have studied in textbooks all these things. So if you divide that, that gives you in crystal clear the so-called progress of life or the margin of life. Similarly, if you look at this, people usually give you the growth in the form of diagrams. Here is one example, primates. As I told you, they originated some time back, and then these are classified into prosimians and anthropoids. Anthropoids into humans, apes, and the so-called monkeys, which are old world monkeys as well as new world monkeys. Apes, orangutan, gibbon, gorilla, chimpanzee, they are closest to humans. And humans are of varieties. The old humans are the human-like apes, and then ape-like humans. All these are listed. Now, all this has taken millions of years for evolution. This is an example for the evolution of horse. We will not go into detail. It simply tells you that life did not come simultaneously for all living organisms. It started as a singular event, and then it diversified by the mechanism that Darwin has told us, and that is descent with change. Darwinian ideas strangely spelled out as theory of organic evolution by natural selection, because his book is titled Origin of Species. Now, speciation is a totally different concept. If you follow these ideas, you will get evolution. Speciation is not by this, it is by some other mechanism. We will not talk about it. Habitat fragmentation and other things is the greatest idea in the history of human thought or philosophy. What Einstein's theory of relativity did to physics, what molecular orbital theory did to chemistry, what the evolutionary ideas of Darwin and Wallace have done to biology. So it is our greatest concept, the only concept that biology has contributed. In the process, slowly, in the early part of 20th century, a totally different approach was taken when people learnt about Mendelian inheritance patterns and genetics started, population genetics and others, then people had different ideas of how evolution can take place. Ron Fisher is a statistician who studied genetics, population genetics. Most of you have studied Hardy-Weinberg law. We will not go into detail. J.B.S. Haldane, one of the greatest thinking biologists, he talked about origin of life, Haldane-Oparin hypothesis. Then this Sewall Wright, who also used extensively population genetics and came with some ideas. Summary of these ideas, this is Ernst Mayr sitting on the right side. And this Mayr is the greatest biologist after Darwin. He can overshadow even Dobzhansky. He was alive for 101 years. Even in his 100th year, he wrote a book. He wrote more than 35 books. His greatest biologist in the contemporary era. Now, all these people talked about another way of evolution, and that is by large-scale mutations. Then these were synthesized, and today we talk mostly of Darwinian evolution, but by mechanisms which involve recombination errors during meiotic recombination, causing changes in the organism, and then perpetuation, and then nature acting as selection pressure. So those who are well endowed will survive and develop. So these are the summary ideas about the classical evolutionary ideas of Darwin and thing. I have not discussed many things, nor about Lamarckism and others, but this gives you a summary idea. However, biologists who watch for this, who believe in Darwin and others, if you look at modern biology textbooks and what modern biology departments are teaching, do you see any teaching with faith, with conviction on evolutionary perspectives in modern biology teaching? You don't see. There are many biology departments in very good universities in the country who don't even have a single paper on evolution. That's a tragedy. Instead of engaging themselves in answering the question of mechanism of evolution, biologists use the term natural selection as an explanation to everything what happens in biology and asked only one question, why things happen that way. The only answer to everything was selection advantage. Hence, biology was hijacked to answer the following questions. What are the proximate causes for physiological and behavioral processes? This is the crux of reduction biology. When we talk of mechanism of hormone action, mechanism of memory, mechanism of immune response, mechanism of digestion, mechanism of growth, mechanism of reproduction, we are talking only answer to the first question. What is the ultimate cause for the ecological and biological phenomena, including evolution, is the second question for which we don't have answer. Nobody is attempting answering the second question. This is unfortunate because people have lost interest. They are only enamored of molecular biology and biochemistry today. Completely fragmented viewpoint on biology. The three phases in the growth of biology, therefore, resulted in explosive knowledge. Two questions remained, however, unanswered. How to understand biological structure and processes? How to explain all this as a manifestation of a fundamental paradigm or concept underlying biological processes and phenomena. Please remember that physics tells you that this material world is real and all that they are talking follows what is called as deterministic model. There is a cause and effect for this, but they don't talk about ultimate cause. Whereas biology tells you that there is no deterministic model in life. It is all following what is called stochastic model. I know you won't understand all this, but we can discuss this later. And therefore, we have a fundamental misunderstanding of what is biology, what is life, how to explain and understand living processes. And that brings us to this, how to teach and how to understand biology. Evolutionary theory with all subsequent modifications has to become the theoretical framework on which we have to teach the entire biology. Please remember, NEP 2020 also recommends interdisciplinary, multidisciplinary nature of so-called disciplines. And in that, only biology will fit the bill, not botany, zoology, microbiology, not physiology, biochemistry, biophysics, genetics, and other things. Biology has to be taught as biology. How to bring integration among the three phases of biology, in vivo, in vitro, and in silico biology, is the biggest challenge in teaching biology. And therefore, we come to this terminology which we will have to understand. I don't have time to go into all this, but let us focus now on this topic of today's seminar, which is molecular evolution. As of now, the topic evolution is taught in isolation as a paper. The syllabus has remained the same, evidences of evolution and selected examples of successful evolution like the horse or human. Kindly remember, 90% of evolution ends up in extinction. No teacher will tell you all this. Reductionist biology has remained immune to discussions on evolutionary aspects. Modern tools to investigate evolutionary phenomena are not taught in majority of places. And so we come to this, what is this molecular evolution or chemical evolution, and how do proteins evolve, which is what we are going to discuss in the next half an hour. How to understand chemical evolution in light of the biochemical unity underlying all living organisms. Please remember, teachers make two contradicting statements. One, that there is a biochemical unity underlying living organisms. Then why do we talk of biochemical evolution? And the second statement is that all proteins, all biomolecules, all chemicals also have evolved. These two are contradictory statements. Diversity of biomolecular composition is very small among animals. If you look at glycolysis as an example of a metabolic pathway. Kindly mute your microphones. If you look at the diversity of biomolecular composition, you see that the diversity among living organisms is very small. You have primary metabolites which are present in every living organism, E. coli or elephant, plants or animals, microbes or others. However, the microbes and plant systems differ from the others in having secondary metabolites. Barring this difference, you don't see any difference among living organisms. So why talk of evolution of all this? All right. It is significant among microbes and plants. We call them secondary metabolites. You have heard about all this, like rubber, pigments, terpenoids. And to talk of evolutionary path for such entities is a formidable task, not yet achieved for even a single group. However, same molecular group acquire different functions during phylogeny. We will give couple of examples for that if you call that evolution. Protein hormones in vertebrates are poisons, snake poisons and others in other organisms or animals is an example. We will take some examples and show this. However, this is another example of that. If you take venoms in snakes and salivary proteins in mammals, you notice a lot of similarity. We'll carefully use this word similarity. One should not talk about homology and analogy for molecules. In mammals evolved from the same ancestral gene, this is what we learn in some group of proteins. Variations in metabolic pathways or physiological mechanisms are never discussed in light of adaptive advantages or disadvantages, but this is exactly what you discuss when you talk Darwinian evolution. A glaring example is parasitology. Parasitism as an evolving relationship, both in terms of mechanisms of survival and as a target of selection, is never discussed in textbooks as well as in classrooms. So there is a lot of misunderstanding of this area of evolutionary biology. This seminar, therefore, should try to discuss the following: the basic phenomena of evolution and conceptual understanding of the same, how to present and discuss physiological, biochemical, and behavioral processes as evolving phenomena subject to selection, discrepancies between conclusions drawn from molecular and morphological evolutionary studies. Of course, I won't go into detail. I'm only pointing out the discrepancies that we face in this area of evolution. Natural selection cannot act if non-Mendelian blending of genotypes occurs, as suggested by Charles Darwin. However, this Darwinian evolution, unlike what he was talking about, that it takes millions and billions of years, we can watch it in a short span of life. That means in a small time scale, and that gives us confidence that same thing must have happened in the so-called Darwinian evolution. As an example, we look at the so-called HIV disease caused by the immunodeficiency causing virus, and a drug called 3-thiocytosine. Most of you know that HIV causes tremendous damage. A lot of work has come, and people invented certain antiviral compounds. One of them is 3-thiocytosine. The structure is shown here. In the initial days, because this inhibits reverse transcriptase, please remember these viruses are RNA viruses, and therefore they have to first make a DNA, and then the following other things happen. And the enzyme that catalyzes this is called reverse transcriptase. It is full of error prone. That is why mutations are so fast in viruses, and this compound inhibits reverse transcriptase. So in the initial days, it inhibited HIV symptoms caused by the virus, and so everybody was happy. But very soon, the virus developed, that means the new mutants develop resistance to this compound. How did this happen? Because the single mutation, a methionine change, essentially made the enzyme reverse transcriptase resistant to this action of this drug, and therefore the new mutant virus essentially did not respond to this compound. Same thing happens in antibiotic resistance in bacterial diseases. We come to another aspect of this, artificial evolution. Proteins, we can demonstrate artificial evolution if you take examples of antibodies and all. Artificial selection can also yield, let us say, example of increased milk yield among buffaloes or cows of different strains, races. We can show that artificial selection can give rise to new phenotypes. Artificial selection, however, cannot lead to speciation by interbreeding criteria. There are 7 definitions for what is a species. One of them is that ability to interbreed. Now, if you take this definition, which is a biological definition, then interbreeding is not banned when you develop new things through artificial breeding, like we do dogs and other breedings by phenotypic criteria. Ring species very continuously vary till new species come. Salamander is an example. Analogy and homology ideas do not apply to proteins and nucleic acids if you have a strict definitions for this. Take, for example, this term molecular evolution. It can be examined, of course, experimentally. And what do people do in this? They take a given functional protein like an enzyme or a toxin or a hormone, isolate it from different species throughout phylogeny, and then sequence the amino acids. This is called the primary structure, which Dr. Yogendra Singh has already told you about, and align these sequences and ask a simple question: what changes are there in the position of these amino acids, some amino acid, among these proteins from different species? And they get a a dendrogram. This is called phylogenetic analysis. And from that, from mutation rate, they calculate for this amino acid to become that amino acid, how many years will take, and so calculate what is called evolutionary distance. Now, the whole thing has no meaning, because if you try to follow this and then compare it with morphological variation, distance calculation, and evolutionary process, and the phylogenetic trees that we get, we see that many a time there is no correspondence between phylogenetic trees constructed out of nucleic acid or protein sequences and phylogenetic trees constructed out of morphological comparisons. You can even have an artificial selection pressure. Let us say in this diagram, you can artificially select for a given RNA with a given secondary structure with a property of ability to bind ATP by a series of affinity chromatography, which Dr. Yogendra Singh has already told you. Therefore, you understand the principle behind. So if you do this repetitive affinity chromatography, you will get an ATP binding RNA with a unique secondary structure shown on the right side. And that can also tell you that evolution can occur at the nucleic acid level. But how does it happen in nature? This is what we have to understand and explain. To make this also a little more clear, let us look at a first order reaction. And all of you have studied a little bit of physical chemistry. You understand all this. And here is an equation which tells you that N is equal to N0 multiplied by E raised to the power of lambda T. Now, lambda is a decay constant. Radioactive decay is a typical example. Evolution also can be taken as a typical example. And T is the half-life. So don't worry about the equation. Based on, if you take a modern sample, a fossil sample like a zirconium silicate, and then calculate this half-lives has passed from origin. Why are you disturbing? Dr. Khan, can you keep all the students shut?
Yeah, I muted them.
I think they don't understand anything.
They are not students. There is somebody from teaching group, I think.
Okay. So based on such a study on a piece of zirconium silicate in Australia, the date of Earth was fixed as 4.5 billion years. This is an experimental way of how do you know human being is so old, or Earth is so old, or a pig is so old. This is the method which is called radioactive dating procedure. All these are helpful in understanding evolutionary process. This is a typical structure of DNA. This is only shown to explain to you the DNA, just like protein has 20 amino acids, DNA is made up of only 4 or 5 types of units, monomers called nucleotides. And these, in the case of DNA, the long chain of nucleotides, what is primary structure, they are linked to each other through hydrogen bonds. You have been told all these details, and that gives you the secondary structure of DNA, what you people call as double helix. So when Watson and Crick demonstrated that DNA has this double helical structure and were promptly rewarded with a Nobel Prize, they never determined the structure of DNA. They only told about secondary structure. Most teachers don't tell this. Most textbooks also are silent on these things. Now let us understand this in a little more detail, especially protein evolution, and understand this from the general background of mutation rate and rate of evolution. Here are 4 examples given to you. An example from the viruses, HIV, human immunodeficiency virus. An example from bacteria, Escherichia coli. An example from insects, Drosophila melanogaster, which fruit fly, which you know very well. And an example from mammals, that is human being. Now, if you notice, the rate of mutation per nucleotide per replication, I said all changes come from replication error. and that you call it as a mutation. I am not referring to cosmic radiation induced mutation. I am referring to only replication error. So if you call this as a mutation and find out the rate, you see there is very little difference among all these living organisms. Only viruses have a little higher mutation, that is 1 in 10,000, 10 to the power of -4. But if you look at all the other organisms, including plants, you have 10 to the power of -9, 1 in so-called billion replications. And that is a very, very, very low rate of mutation. Kindly excuse me for 2 minutes. I'm sorry for the interruption. I have a hukum from my wife. That's the problem. Okay. Therefore, you see that the rate of individual mutation expressed as per nucleotide per replication is more or less similar. However, rate of mutation or evolutionary change, you notice there is a vast difference among all the living organisms, as is given in the last column. This because the sizes of the genomes are different. A virus has hardly 10,000 nucleotides in its genome, whereas if you take Escherichia coli, it runs into about 2 million. Drosophila, about 3 into 10 to the power of 8. And human being, you all know all this. I'm talking about diploid organism, therefore 6.6 into billion. And what is most important is how many cell cycles or replications come between generations. In the case of viruses, it is only one. One replication, it becomes another virus. In the case of E. coli, the same. Whereas in the case of Drosophila, during gametogenesis, there are about at least 20 cycles. And in the human, in the case of man, it's about 200 cycles, spermatogenesis, whereas in the woman, egg formation gametogenesis is about 33 cycles. And therefore, if you take into account these things, the mutation rate per genome, per generation changes in all these organisms from 1 in viruses to 10 to the power of -3 in the case of bacteria. These are all average figures. 4 in the case of fruit fly and 200 approximately in the case of human beings. And therefore, there is an enormous difference between rate of mutation per nucleotide and the rate of evolution as we see in different organisms. However, we have a conceptual problem here. What is the biochemical description of homology and analogy? No textbook will tell you. All that they will talk is similarities in amino acid position in different proteins, same protein from different species, and how these amino acid changes are brought about by mutation. What is the embryological origin in biochemical language? We have not understood. Structural similarities do not mean homology. It only means similarity or even same function. I'll give some examples to make you clear. Phylogenetic distances are not same for all proteins or genes, nor match always with morphology-based phylogenetic distances, a point which I have referred to earlier. Single nucleotide polymorphism, which most of you read in modern biology, in a given population, they do not indicate common ancestry, as in the case of Darwinian evolution. So there is a tremendous misunderstanding when you talk of protein evolution and the classical Darwinian evolution of whole organisms or whole populations. Dendrograms or molecular trees are okay for microevolution. They are not okay for macroevolution. Your fossil evidence or paleontological evidences talk about macroevolution, large-scale changes. We have a person called Stephen Gould who also gave this idea that evolution is not always gradual like Darwin envisaged, that many a time there is a quantum jump, huge changes occur, which we call as macroevolution, when you see classes of organisms appearing or disappearing. In the case of the so-called protein evolution, therefore, we don't understand natural selection and other things which your textbooks are talking. So to make us understand, a Japanese scientist called Kimura floated an idea called neutral mutation theory. And by this, he means that mutations have no directions or purpose. All mutations are neutral. What happens is the consequences of the mutation is only drift. There is no natural selection, there is nothing. They just drift and you see a new change. Now, this he was referring to in the case of protein evolution. It's a difficult idea to understand. However, quickly people misunderstood this Kimura's ideas and they applied it to populations and organisms, and their hell broke because this is not applicable to population evolution as Darwin was talking. You can't talk about neutral mutations because every mutation that occurs in the Darwin idea can be neutral, but always it has a certain consequences, which if selection eliminates, then that organism does not change anymore. It is gone, it is extinct. However, if it has a selection advantage, that organism survives and breeds more. And therefore, that's how evolution occurs. However, therefore, this neutral mutation theory, which causes a drift, is essentially for protein evolutions and not for organism or population evolution. This is a typical structure of a protein like myoglobin. You understand this as a tertiary structure of a protein, which Professor Yogendra Singh talked about in the morning. Let us look at some other changes which completely confuse us. If you take myoglobin, one of the muscle proteins, and look at the sequence of this taken from chimpanzee and compare it with that from a human being, chimpanzee is as different from human as possible, but the myoglobin sequence changes only 1 in 153 amino acids. That means one single amino acid is different between chimpanzee and human, and you can't conclude that that's how a chimpanzee became a human. Impossible. That means molecular changes cannot be projected as an individual mechanism and calculate evolutionary distances, because this will never superimpose on the population-based morphological changes-based evolutionary trees. This is a typical example which will create hell of confusion in students. You look at this so-called ribonuclease as an example. It's an enzyme. It's a digestive enzyme. You look at this same enzyme from cattle, bovine, from humans, and you notice that there is not much of difference. Similarly, if you compare, these are called orthologs. If you compare the same ribonuclease in a human being from another protein unrelated to ribonuclease called angiotensin from the same human species, you see there is a lot of similarity. This is called a paralog. And human angiotensin does not do the same thing, same function as ribonuclease. But how come they have the same structure? That is the confusion. How proteins evolve, it doesn't seem to be discussable from the Darwinian ideas. Same another example I have given. And therefore, giving rise to phylogenetic trees, as many bioinformatic people construct, taking a protein example and then taking it from different species and then aligning them and calculate the distances, you will get trees like this. But what is that we make out of this? We don't understand. Same thing applies to another functional protein, which all of you are familiar. What is enzyme evolution? most of you remember. I'll give you another lecture on enzymes later. But right now, you please remember enzyme is most of the enzymes are proteins. And the enzymes have active site. It catalyzes the conversion of a substrate to a product. And they have two properties. One is called affinity, Km. Another is called maximum velocity or catalytic turnover. If Km values of an enzyme from different phylogenetic groups is compared, we do not see any direction towards higher or lower value when superimposed on organismic phylogenetic tree. This is a classic example of a discrepancy between evolutionary tree constructed out of enzyme evolution, evolutionary tree constructed out of population evolution. Km value and catalytic efficiency have no correlation. Kindly note this, kindly remember this. Most efficient enzymes have very large Kms, which is difficult to understand. And if the Km is low, which means affinity is very high, there are many inefficient enzymes with that kind of a thing. Therefore, we do not understand when somebody says enzymes have evolved. Have they evolved towards catalytic efficiency? What changes are these correlatable with affinity? Therefore, variation in the Km value can be indicative of random and directionless change, and so Kimura becomes correct. This is fine only as far as protein evolution. Don't apply this to organismic evolution. But what is the adaptive significance of Km value is very difficult to comprehend. Like we discuss in the case of Darwinian evolution, we talk of adaptive significance of a given change and therefore survival value, survival advantage. But this significance, if you talk about for proteins, especially an enzyme, say a functional protein, we don't understand the correlation to reproductive fitness, which is the ultimate thing which leads to breeding and production of the next progeny, and through which alone natural selection can act. And therefore, you see a difficult concept, this protein evolution, and how to understand it from the Darwinian classical ideas of evolution. Now, I wish to stop at this because it's a very, very difficult concept, and I would like more discussion on this before we disperse for lunch. Thank you, Dr. Khan. Thank you for the audience for your patience. I want more discussion on this because it's a very difficult concept.
Thank you so much, sir. One question I have got from YouTube subscribers. There are 2-3 questions actually. One thing is which text we use for getting material on this concept? What are the textbooks or what is the literature is a question.
I'll give you a joke. There are a number of books on molecular evolution. Okay? Stalwarts of evolutionary biology have written this. These are available in any good library. I'm sure your library also has. And the man who talked so much on this, Kimura, he himself does not believe in the neutral mutation theory. It is others who are popularizing it and talking about it. So this area of evolutionary biology, especially molecular evolution, is full of confusion created by senior professors, great textbooks, which you call, and great libraries. That is why I said you need a critical discussion about all these things. Teachers don't tell these things when teaching a class. They talk from books which are about 30 or 40 years old. They're not aware of the current thing. That's why academies are starting all these workshops. There are a number of books. I'll give you the whole list later by email. Okay.
Sir, second question by same person is, how is macro evolution measured?
Yeah. Macroevolution is the typical Darwinian evolution of, in terms of not changes in species, it changes in genera level, changes in class and other orders. And therefore, the only evidence for this is the paleontological evidence, which every teacher will tell, every textbook will write. But how do we understand this in terms of mechanism? The only idea that we have is the Darwinian idea of natural selection, but the details are known only for 3-4 animal groups, or maybe flowering plants in case of plants. We don't, or from water living aquatic organisms, how did they become trees or plants or land organisms? There is a little bit of data on these things. We can discuss this later, but otherwise, macroevolution is very, very difficult to explain, because one is how many such events have to occur before one species becomes another species. In fact, the biggest problem in this evolutionary biology is the mechanism of speciation. Only habitat fragmentation can explain this. But Darwin's book title is Theory of Organic Evolution by Natural Selection, and he said he is talking about speciation. Speciation is a different concept. All right? That's why it is difficult to understand mechanism of macroevolution. Microevolution, protein evolution, we can understand, but this evolution that we are talking is only in the dictionary meaning of this word, which is change. Changes in amino acid sequence, changes in nucleic acid sequence, we can explain due to mutation, but how is it related to selection? How is it related to reproductive competence? All these things, books are highly silent on this. Okay.
In continuation, the question has, do the macroevolution involve mutations or frame shift or any other kind of mechanism?
Yes, yes. Everything is due to mutation. But when we talk of evolution from Darwinian concept, of course, he himself did not know the origin of mutation. Subsequent developments in genetics told us that major part of the mutation is during changes in the recombination, meiotic recombination. That's where the errors come. However, there are other mutations which can occur, chemicals-induced mutations or cosmic radiation-induced mutations. But unless they happen in the germ line, they don't have any meaning to discuss evolution on this. It only affects ontogeny in the individual life organism, but not their progeny, because progeny can be changed only through gametes, fusion, fertilization, and the reproductive process. And therefore, any alterations in the adult organism in somatic cells have no meaning. So the only mutation we relate is the recombinant errors that happen. Of course, Darwin didn't know all this because genetics was not there at that time. Even Mendel, when he formulated his so-called laws of inheritance, he himself had serious doubts about it. He corresponded with Darwin. Darwin simply did not believe anything that Mendel was talking. He tore the letter. You can check with the history of biology, all these things. That means great scientists also can make mistakes. And therefore, summary is that, yes, mutations are the primary cause of change. And if this happens in germline, therefore recombination errors, then it can be passed on to the next progeny. And out of these, the so-called physiological consequences of such mutations, if it affects reproduction, then selection pressure will eliminate them. If it does not, then those organisms or populations are selected, and therefore you see the process of evolution. That is the explanation.
Sir, one more question. Maybe it is related to this or not, but the same participant has asked, there is no correlation between Km value and catalytic efficiency.
Yes.
When we consider the enzyme evolution, can you highlight this point?
Yeah. See, if you take enzymes as an example, we will understand this after the 4th lecture. You have two properties. One is called maximum velocity. That means the catalytic efficiency, where it catalyzes maximum rate of the reaction if the substrate is at a saturating level. And the second property is Km, which is roughly translated in English as affinity. It is not literally affinity. It is much more than that. No textbook of biochemistry will give you the proper meaning of the word Km. Let us loosely translate as affinity of an enzyme for a given substrate. Okay. Now, when people talk evolution of protein, what they mean is changes in the amino acid sequence of a given enzyme. Compare it for the same enzyme, but isolated, let us say, throughout vertebrates. If you isolate the same enzyme from fish, from birds, from amphibians, from lizards or reptiles and mammals, take 100 examples or 200 examples, and then align the sequence, you see some lot of amino acid differences. If you call this in English, you should call it a change, but people use the word evolution. Now, if you look at this change and calculate, there are ways of calculating distances and all that. You can ask now the question, given enzyme from different vertebrates, these are the changes happening, but what is the direction of this change? Is it towards catalytic efficiency? Has it got any correlation with Km? Can you say that if you have a Km increases, you have more efficiency? No. Can you say that if Km decreases, you have more efficiency? No. Therefore, we don't seem to understand what exactly is enzyme evolution, towards what. But if we understand that evolution means only change, yes, enzymes have changed. There is no question on that. There is no doubt on that. But to use the word evolution for that, I think is asking for impossible, because all enzymes are not equally efficient. What you see today are the evolved enzymes, which means selection pressure has already acted and they are surviving. Therefore, you can't say that these are all evolved in that sense. The same thing holds good for even macroevolution. Evolution to what? There is no direction for evolution. Please remember, you can only quantitate the changes happening and calculate distances. So in the case of proteins, especially functional proteins like this, or even hormones, we will not discuss that now. There doesn't seem to be any relation, correlation it is called, between Km value and catalytic efficiency, which means at a given substrate concentration equivalent to Km, what is the velocity of the reaction? Do you see this more with Km low? Do you see this more with Km high? Absolutely no answer. There is no correlation. That is the point which I am making.
Sir, one last question has been asked. Explain all enzymes are proteins, but all proteins are not enzymes.
Yeah.
That is actually in next lecture, actually, but the participant has asked now.
Yeah, that's okay, no problem. See, when people discovered metabolism and other things and came to a general conclusion that there is no reaction within our body or any living organism which is an uncatalyzed reaction. Unlike in general chemistry, there is no uncatalyzed reaction. And they were asked a question, how are they catalyzed? They discovered enzymes. It so happens from the discovery of the first enzyme till 1980, all the enzymes that they discovered happened to be proteins. So a general statement was made in biochemistry textbooks that enzymes are all proteins. Of course, proteins can be hormones, proteins can be antibodies, but proteins can also be enzymes. And so you got a paradigm that all enzymes are proteins. This is a statistical error. We will discuss this in some other lecture, how badly we conclude and make errors. This is a stochastic process. Whatever it is, they made a general statement, enzymes are proteins. In 1980, a Nobel laureate called Thomas Seck, he got Nobel Prize for this only. He discovered that ribonucleic acid, ribosomal RNA, has catalytic power. What does that mean? It can catalyze without any protein contamination some chemical reaction. And therefore, he said, some ribosomal RNA have enzymatic property, but they are not proteins. So they called it ribozymes. Somebody else after few years, discovered that antibodies to certain compounds, even though they are proteins, antibodies are only immune reactive proteins, but some of these antibodies against reactive intermediates in enzyme catalyzed reaction becomes enzyme, which means antibodies, certain antibodies become enzyme. They have catalytic power. So these are called abzymes or antibody enzymes. Like that, we can go on. Today, we know that even naked DNA has enzymatic activity. Therefore, we make a general statement typical of all classroom lectures that all enzymes, earlier they said all are proteins, but all proteins are not enzyme. Proteins are also hormones, antibodies, and all. Similarly, we cannot even make that all enzymes are proteins because there are abzymes, there are ribozymes, there are single compounds like histidine can have catalytic power. And therefore, we should not make any final statement during teaching or during learning because science is characterized by dramatic changes in our understanding in concepts. That is the warning I am giving through this lecture.
Thank you so much, sir, for answering the question.
Did I make myself clear?
Yeah, sir. The replies they are replying the participants who have asked questions. They are replying the satisfaction.
Very good. Because I know it is very difficult and normal teachers don't tell all this. They don't clarify anything. It is information transfer. And that is where all the damage has been done to higher education. TK, all right now.
Okay, sir. We have now come to an end of the session 1 and session 2 of the day 1. So we will be joining back at 2 o'clock sharp for the next lecture, session 3, where Professor
Yogendra
Singh sir will be joining with us.
Yeah.
So we'll have a lunch break and sharp 2 o'clock we'll be joining in the meeting.
So I can go off now and again enter at 1:55 or so. Is it okay?